So thanks a lot, Robert, for great introduction and for the invitation to come here. I greatly enjoyed my time in Vienna, and it's been a wonderful visit so far. And I had a lot of very interesting conversations. So let me just briefly guide you through the outline of my talk. Uh, and first, I'm gonna make a very brief introduction about the things that my lab is working on, which is horizontal gene transfer and transposable elements, and our primary experimental system, Deloitte rotifers. And then I'm going to tell three different stories, which in one way or the other are connected to horizontal transfer, transposable elements, and Deloitte rotifers. So uh, to begin with, horizontal gene transfer is the means of transmission of genetic material that is non-vertical, meaning it doesn't go from parent to offspring. And it's the predominant mode of transmission of genetic material in bacteria, but it's not nearly as ubiquitous in eukaryotes. In fact, it's very rare. Their germ line is usually fairly well protected from external DNA. So the role of horizontal transfer in eukaryotic evolution has been hotly debated, and some people believe that it doesn't play a role in evolution, but when we encountered massive horizontal gene transfer in Deloitte rotifers back in 2008, we thought that it would be an amazing eukaryotic system to study the role of horizontal gene transfer in eukaryotic evolution, because we found that it occurs on a massive scale and from highly diverse sources. Like, we detect all possible kinds of non-metazoan sources, such as bacteria, fungi, protists, plants, you name it. And not only the genes get in there, but a lot of those genes, some get pseudogenized upon transfer, but some definitely become adapted to the new hosts, and each in many individual cases which we examined, they serve special functions, which some of which I'm going to tell you about. But first, let me introduce you to Deloitte rotifers. It's a These are microscopic freshwater invertebrate animals. They're a fraction of a millimeter in length. You can barely see them with a naked eye. They're highly resistant to desiccation and ionizing radiation. No males or conventional meiosis have been demonstrated, so they mostly reproduce clonally. Uh, and they live in ephemerally aquatic habitats, which stimulate those frequent cycles of desiccation and rehydration, such as puddles of water, bird baths, like moss on trees. And they're fairly complex animals. About the complexity of C. elegans, I would say, it's animal. The animal has about 1,000 nuclei, but um, it's a fairly complex system of organs, and you can see those bilateral ovaries here, and the characteristic corona, which helps them to feed. They basically sit all day filtering microscopic particles through it. And in the metazoan phylogenetic tree, they're, I would say, about as distant from humans as are the best studied eukaryotic model organisms such as Drosophila and C. elegans, but they don't belong to ecdysozoans. They belong to these more wormy-like creatures in the Lophotrochozoan, like this, somewhat close to flatworms, but they make a pretty big phylum Rotifera. The phylum consists of two major classes, and one class is called Monogononta. They reproduce facultatively sexually, and there's about 1,500 species in it, but we primarily study the class Deloidea, which reproduce clonally, and it's now nearly 500 species, so it's a fairly speciose taxonomic group. Why do we study them? Like I said, we're interested in their unusual biological properties, such as clonal reproduction and resistance to desiccation and ionizing radiation. Of course, a lot of advantages that they're not model organisms, so not too much is known about their biology, and there are always new things to be discovered. It also comes with a lot of disadvantages because we cannot currently do any genetic manipulations with them. So far, they have been pretty refractory to it, and they're also syncytial, which would complicate things like single cell studies. There's quite a few sequenced genomes, so that's a valuable resource. So we are mostly interested in studying the fascinating aspects of their biology, such as massive horizontal gene transfer and some really weird transposable elements that we find in there. And last but not least, they're exceptionally photogenic. So this picture shows you like each of those shots at some point was awarded by a prize in a photomicrography beauty contest. And so they look really nice under polarization microscope. And this is essentially how a rotifer spends its day, feeding on bacteria. They live on fresh spring water, and we feed them E. coli. And it's just mesmerizing to watch under the microscope how it's moving around. Oh, finally caught up. I don't know why it's such a delay, but anyway, I think it's a very well-fitting soundtrack. So relaxing. Anyway, they're exceptionally hardy animals, and they survive in very harsh environmental conditions, such as Antarctic glaciers and hot springs, acidic rivers, and in Siberian permafrost, they have been frozen for 24,000 years and then defrosted and started swimming like nothing ever happened to them. So that was really fascinating study that was published a couple years ago. Uh, anyway, our contribution to rotifer biology was that we, back in 2008, discovered massive amounts of foreign DNA in their genomes by cloning big chunks of genomic DNA. We tried to focus on subtelomeric regions, and we found a very heterogeneous mix of metazoan genes that would normally be present in any metazoan genome, but also a large number of genes from various non-metazoan sources, such as plants here in green, bacteria here in blue, fungi in purple. And like I said, the metazoan, the core genome regions look pretty much metazoan, very densely spaced metazoan genes with relatively short intergenic regions. But when you get to telomeric, subtelomeric regions, this is where you see most of horizontally transferred genes, and also a large number of transposable elements of nearly every possible kind: DNA transposons, LTR elements, non-LTR elements. So let me remind you that there are two types of transposable elements. Class 1 are retroelements that need RNA intermediate for transposition, and they encode the enzyme reverse transcriptase, which makes a cDNA copy of an element and moves it into a new genomic location. And class 2 DNA transposons, they encode a DNA transposase, which basically excises a copy of transposable element from one genomic location and then inserts it into another location, usually leaving behind a double strand break, which needs to be repaired, of course. Well, these are like more specific classes of transposable elements. We are detecting most of them in deltoid rotifers. They have a very high diversity of transposable elements in their genome. Practically the only type of elements they're missing is those maverick pollenton elements. We cannot find those, but any other type of mobile elements, they're present. Uh, there is a large variety of families. We could count about 260 different transposon families. But the characteristic feature is that they are present only in very low copy numbers, like 1, 2, 3 copies per genome. So they never amplify to high numbers in the genome. That was also an interesting feature of their biology, because and it also creates big problems with transposon annotations, because most of the software that's looking for transposable elements is trying to find some multi-copy sequences. So all our repeat libraries had to be carefully manually curated to make good quality libraries. So we were mostly interested why is the transposon content in rotifers so low. As you can see on this scale, on the eukaryotic scale, it's even lower than in yeast. That's like most of the metazoan genomes have a relatively big chunk of their genome occupied by transposon insertion. So there was quite a it seemed a bit contradictory with their ability to horizontally acquire different genes from non-metazoan sources, because about 8 to 10% of all coding sequences are represented by foreign DNA. So how do they keep their transposon content so low? Well, of course, first we looked at small RNAs that they have most of their small RNAs are quite abundant, and about 65% of those map to transposable elements. Some are also mapping to genes of foreign origin. A very unusual feature of their genome was that they amplify to a very large extent the genes that are responsible for pRNA mediated silencing. Like the first genome sequence revealed that they have 15 copies of PVs and argonaudes. And later, when other delloid species were sequenced, the number of those genes went even higher. Like for RNA-dependent RNA polymerases, it can go as high as 20, 40, 50 copies. Some really crazy numbers, but if you compare it with monogonant rotifers, there is clearly a sharp contrast because they have very few of these genes. And the class monogonantite doesn't control the transposons as well, because the transposon content can vary anyway between 14 and 45% in various sequenced genomes. So they use RDRP instead of ping-pong mechanism. And another curious feature is that the pRNA response is initiated even for single copy transposable elements. So we were interested to know if they in general have particularly efficient genome defense mechanisms against transposons, and from just looking at various ways in which they can get rid of their transposons, we were kind of seeing that they're fighting their transposable elements in any way that they can. Like a lot of transposons get damaged by microhomology mediated deletions. Then there is a very strong trend to recombine between LTRs of each LTR retrotransposon to pop out the internal part so that there would be only one LTR left behind. And like I said, they do post-transcriptional silencing and transcriptional silencing. And more lately, we've been interested in epigenetic mechanisms of their suppression, such as DNA modifications. And of course, this is not present in Drosophila, but in rotifers, we also were looking for DNA modifications, and as in Drosophila, it doesn't have the mammalian type of DNA modification, and which is present in most eukaryotes, the 5-methylcytosine, which is the most common form of DNA epigenetic modification. So my first story deals with the bacterial methyltransferase, which was transferred from bacteria into rotifers. And it was very unusual to see it in the genome because it contained this very characteristic part of a bacterial methyltransferase. But we knew that it's not a contamination because it has accumulated introns. We had 2 allelic copies in the genome, and they were surrounded by proper metazoan genes. And what's very important is that it was fused to a eukaryotic chromodomain, which typically recognizes silent chromatin marks on the H3 histone. So this is just to briefly remind you the what kind of DNA methylation is out there. Like I said, most eukaryotes have 5-methylcytosine, and most recently 6-methyladenine was also detected in eukaryotic genomes, although its role is still controversial. But in our case, we detected this 4-methylcytosine, which has not been found in any eukaryotes, but it was found only in restriction modification systems of different bacteria. So at some point in rotifer evolution, this horizontal transfer event happened from bacteria into rotifers, and this chromodomain has been acquired very early in their evolution, because we see this same configuration in all members of the class Deloidea, which are like 3 different families, very divergent. And the rest of the tree here is represented, well, it's not a phylogenetic tree, but it's sort of clustering by sequence recognition. And these are all from bacteria, and it mostly clustered with 4MC rather than 6-methyladenine methyltransferases. So we assumed that it would probably recognize 4-methylcytosine. So we inspected all the candidate methyltransferase that were present in the genome, and our candidate was the only one that was not present in other eukaryotes. So it was present only in Deloid rotifers and in bacteria, here in red. We found like a candidate methyltransferase that might methylate adenine, but these candidates were later discarded because they were annotated as DNA, but were later shown to methylate proteins. So we focused on this methyltransferase fused to a chromodomain, and our preliminary experiments detected that the pattern was consistent with it being 4C methyltransferase, because it was being digested by methylation sensitive restriction enzymes in a way that's consistent with 4-methylcytosine. And also when we used the antibodies against 4-methylcytosine in a dot blot, we could clearly detect the signal, which was not present in the bacterial food. There is also some methyladenine in the genome detected, but we focused on expressing this methyltransferase as a recombinant protein. So when we introduced it into E. coli and overexpressed it, sure enough, it was capable of adding 4-methylcytosine to the substrate in vivo and in vitro. And when we mutated the catalytic residues, it was no longer doing that. So it was clearly responsible for catalytic activity. Not surprisingly, when we introduced it back into bacteria, the chromodomain was not important because it was working even without it. But in any case, we wanted to see what's going on in the eukaryotic system and not in bacteria. And we ran a series of experiments to detect that the 4MC mark is located at transposable elements and certain tandem repeats. And we analyzed it by complementary methods. We used the less sensitive method, which involves antibodies. But since we had those antibodies, we did this so-called deep sequencing, and which stands for the DNA immunoprecipitation. And we also used PacBio smart sequencing, which is particularly good at detecting bacterial DNA modifications, such as 6-methyladenine and 4-methylcytosine. It's pretty bad at detecting 5-methylcytosine, but in this case, we didn't need it. So after running some assays, we saw that mostly the mark is localized at 5' boundaries of transposable elements, which is where the promoter is located, and that it's also dependent on transcription, because we didn't see any signal from short copies, but only from full-length copies or from some copies that were incomplete but still preserving the promoter at the 5' end, so they were transcribed. And when we examined transcription, we saw that it's the RNA-seq data show reduced transcription of transposon genes which display this 4-methyl-C mark. And there was correlation with small RNAs, and also a very good correlation with histone modification marks that are determined by ChIP-seq against with antibodies against the silent modification marks on histone. And finally, when we searched through the genome, we found that rotifers have greatly amplified this particular histone methyltransferase. It's in most species, it's present in single copy, but in rotifers it was present in 7 copies, and one of the variants developed preference for MC on DNA, which basically led us to propose this model that this methyltransferase can recognize silent mark on the histones and then move it, deposit 4-methylcytosine onto DNA, which in turn would be recognized by this methyl binding domain on the histone methyltransferase, which would create basically this self-reinforcing loop. And it's known from plants. I don't have to go through the systems of DNA methylations that are present in plants and animals, but I would like to focus specifically on one which is called chromomethylase. It's present in plants, and it works essentially the same way, but it's a 5-MC based by 5-methylcytosine methyltransferase, which contains a chromodomain, and it also has a partner which recognizes the 5-MC mark, and there is a recognition domain, and then it deposits histone silent chromatin marks on the histone, making this self-reinforcing loop. So this is an interesting case of convergent evolution. Does it happen? Did it happen anywhere else? It was also found in a liverwort, Marchantia, but in this case, it has not acquired any extra domains. It was also horizontally transferred from bacteria, but in fact, it already all the transposons here are already covered by 5-methylcytosine. So in this case, it goes everywhere except transposable elements. So it's just the opposite pattern that that is seen with this horizontally transferred methyltransferase. So this is just to summarize our findings of this 4-MC methyltransferase, that it's targeted to transposon and tandem repeats. It acts to in vivo and in vitro, and in vivo it silences transposable elements by integrating into the chromatin system, and it complements the repertoire of genome defense system against transposons in rotifers to provide more efficient protection, because as clonal animals, they should be able to suppress their transposons because a clonal lineage is essentially stuck with its own transposons. So they have to suppress it to prevent it from going extinct. So another case of horizontal transfer was unexpectedly discovered during studies of fungal infection. And this is a collaborative work with a group at Oxford University. So essentially, when you infect rotifers with these fungi, it can be pretty devastating. On the right, you can see what happens to a rotifer when it's infected by this fungus. So it basically consumes the animal in its entirety. And RNA-seq analysis of samples taken 7 and 24 hours post-infection revealed, of course, differential gene expression. And of the two species that were studied, Ednita ricci is much more resistant to fungal infection. And in Ednita vaga, the survival rate is much lower. And correspondingly, Ednita ricci has a much higher number of upregulated genes going up in response to fungal infection. And interestingly, when those genes were analyzed for enrichment, the more resistant species, Ednita ricci, had a very high degree of enrichment for these several categories, which are all correlated with non-ribosomal DNA synthesis. And this was not observed in abiotic stress response, which is like basically response to desiccation, but it was very pronounced in the response to fungal infection. So when those non-ribosomal peptide synthetases, phylogenetic analysis shows that they're undoubtedly of bacterial origin. And those are like very big multimodular enzymes, and each of those modules is responsible for addition of a single amino acid, but they can be, those clusters can be very extended, like in length, occupy dozens and in some cases even hundreds of kilobases, depending on the product that they're generating. So in this case, in Ednita ricci, we could see a tenfold increase in those expression of those non-ribosomal peptide synthetases. And when these genes are mapped on the rotifer genome, there is a fairly good correlation between their location in the subterminal regions of the chromosomes, where also the transposable elements are concentrated. So this next picture highlights the telomeric location, and for one of these products, which is particularly overexpressed, we did prediction of the online of those very complex compounds that they may be synthesizing. It has some common features with some known antifungals, but of course, no one has analyzed those. It would be very interesting to do this kind of metabolomic analysis, but unfortunately, these compounds may be too complex because there may be a lot of modifications added to it. So it's not as straightforward as we would like it to be. But nevertheless, we see that upon challenge with fungal pathogens, horizontal transfer genes are being upregulated very strongly, at least twice as strongly as usual resident genes, and the most overrepresented are those subtelomeric clusters of bacterial polyketide and non-ribosomal peptide synthetases. And finally, I would like to give you some more insight on the what's happening with transposons and telomeres and how it may possibly be related to horizontal transfer. And this, and for the most part, it's just speculations, but there we'll see how what else we can learn about it. So in particular, I'm going to focus on this relatively obscure group of mobile elements, which are called Penelope-like elements. And back in 2003, we have shown that these form a sister clade to telomerase reverse transcriptases. And like when you look at commonly accepted classification of transposable elements, there are distinct groups. Some people assign it to line elements or to non-LTR transposons. In a sense, that's technically correct because they, on one hand, they don't have LTRs, but then they form a very complex structures. Occasionally, it includes the same element repeated in tandem, which can be sometimes mistaken for an LTR. But phylogenetically, they're a very distinct group because they are associated with this special type of endonuclease for their intrachromosomal mobility. It's called the GIYYIG endonuclease, and it can make a nick in the chromosome so that the element could insert in the new chromosomal location. But this is true of canonical Penelope-like elements, and we were mostly interested in those elements that have non-canonical structure in that they do not have the endonuclease domain. And one interesting feature was that they usually carry a reverse complement of telomeric repeats at the 3' end, and they don't have the endonuclease domain. And they're found not only in rotifers, they're also found in some fungi, some plants, and some protists, but they're not exceptionally widespread. So we were looking at, when we were looking at genome sequences, those elements seem to be covered in its entirety by piRNAs, so they're mostly silenced. You can clearly see the boundary that it's a transposable element and that the adjacent genes are transcribed, and these things are generating lots of piRNAs, and they form a typical structure which is also characteristic for line retrotransposons, which is the fixed 3' end and the frequent 5' truncation pattern at the 5' end. So this is very similar to what is found in other non-LTL retrotransposons when the 3' end is defined, but 5' end is often variable. So what we see at the transposon telomere junctions is that it usually has those short stretches of telomeric repeats, which are indicative of their transposition mechanism, that they somehow employ those telomeric repeats to attach to this chromosome ends, which can get exposed. So among other Penelope-like elements, you can really see the difference between canonical elements, which are relatively short and contain either one or two open reading frame. So the reverse transcriptase moiety is here. But for those giant retroelements, here it's aligned with the reverse transcriptase domain. And some of these elements have a lot of introns, some do not contain introns, but they are also characterized by a large variety of additional open reading frames, generally concentrated in the 5' part of those giant retrotransposing units, and they can be as long as 40 kilobases in length. So those are really giant and unusual elements not seen in any other species. So about those additional open reading frames that they encode, they are usually all in the same orientation. Some contain introns, some do not. And there is quite a few additional enzymatic activities which are all optional. They're not seen in every element. Some and exonucleases, endonucleases, esterases, and some putatively structural elements which uh, may lead to a suggestion that at some point they might have some might be forming some structured particles, but those would be really challenging to detect, but it would be really interesting if this is indeed the case. So this kind of summarizes their coding capacity, that they all contain the reverse transcriptase. Some also contain non-catalytic forms of reverse transcriptase, and additional open reading frames of enzymatic nature, which are always optional, and those supposedly structural ORFs, which are not really similar to any known open reading frames, except that they are short. Some of them contain like zinc fingers, some contain transmembrane domains, but in any case, they would be too short to form like canonical envelopes. But also an interesting feature is when we did an inventory of long terminal repeat retrotransposons, we saw like a very similar set of additional open reading frames, which can be found downstream of the Pol gene, which codes for reverse transcriptase. So we also see some endonucleases, some esterases, some transmembrane protein. In one case, we found a real envelope gene which was captured from paramyxoviruses. So it's marked here in green, but that's only one lineage of LTR retrotransposons. So one more interesting feature is that they have these interesting self-cleaving ribozyme elements, and in this case, they're invariably located at the 3' end, so they form probably like this special structure that keeps those telomeric repeats sufficiently open to anneal to the exposed G-rich regions, but we don't really know their functional significance. They're not exceptionally active as ribozyme. We have to keep them for like in very high magnesium concentration to keep to perform self-cleavage, but nevertheless, they're almost invariably found in this position next to reverse complement stretches of telomeric repeats. And when we examine foreign DNA that was present in subtelomeric regions, we also see a high concentration of those short interstitial telomeric repeats. Sometimes they are found like in joint orientation, which makes us think that those segments can just be added to the ends of the chromosome. Like there is if you compare foreign genes and metazoan genes, there is a much higher frequency of telomeric repeats to be found near foreign genes and transposable elements than metazoan genes. So there is definitely some correlation between telomeric repeats and DNA acquisition. And there is a very strong bias towards those telomere-specific retroelements and also foreign genes which exhibit small RNA coverage. So in summary, we're speculating that terminal addition of retrotransposons and terminal addition of foreign DNA may have some features in common, in that they both need exposed telomeres, but we cannot also exclude that transposons somehow facilitate that, especially if they're capable of forming some kind of particles. We don't see those foreign genes internalized within transposons. We usually see them between transposons. So there is no direct capture that we can observe in this case. So this just summarizes the final part about the giant transposons, that they are units forming very long head-to-tail chains on deltoid telomeres. They have frequent 5' truncations and very diverse coding sequences, which are nevertheless optional in most cases, including extra enzymatic functions, putative structural proteins. They're not present in monogonal protophores. They are only found in deltoids. So we speculate that there may be something about these elements that may facilitate horizontal transfer. So about the questions that we would like to study in the future, of course, we're interested in how still the mechanisms, how this horizontal transfer is happening. And of course, it's very hard to look at it experimentally. And there is also some mysteries about their genome, is why they're so refractory to accepting any foreign DNA under experimental conditions, when they can easily, apparently relatively easily, do it over evolutionary time. And those giant retroelements, of course, are still very interesting to explore, except that obviously it's not of very broad significance, because we don't see those giant units elsewhere other than deltoid rotifers. There are some analogs in fungi, but they don't have extra open reading frames. So, and of course, we still need the genetic tools. We would like to learn more about those substances that they produce, because we detect about like 40, 50 different biosynthetic clusters in subterminal regions, which were captured from non-metazone sources. And it's only 2 of those clusters that participate in response to fungal infections. So it would be interesting to know what all the others are doing. And in summary, I would say that studies of non-model organisms can occasionally be very frustrating, but can be also be very rewarding, depending on what you find in there. And of course, I would like to point out that most of this work was done essentially by 2 people in the lab. Fernanda Rodriguez is responsible for high throughput sequencing and data analysis, and Irina Yushanova is my chief biochemist, who is doing protein expression, purification, and all kinds of in vitro and structural analysis. So we had collaborators at Oxford University, and a lot of summer undergraduate students, which come every summer to do projects in the lab and stay there for 2 or 3 months. And sometimes contribute quite a lot to our ongoing projects. And finally, I would like to, if your research has anything to do with transposable elements, don't forget that there is this journal which specializes on mobile elements. So if it has something to do with transposons, you won't get a desk rejection for sure. So, and thank you for your attention.